This is a production of Cornell University.
Did you know that there are fish that communicate and navigate using electrical signals? Professor Carl Hopkins of the Department of Neurobiology and Behavior will show us.
Did you know that this fish that I'm showing you is capable of generating electricity? It has a little battery in its tail. It's an electric fish from Africa, and I'm going to demonstrate that by taking the fish and putting it in this tank over here that has an electrode in it, and we're going to connect the electrode to a loudspeaker so that we can hear the electric discharges. So take the fish out and put him over here in this tank, and then I'm going to turn the loudspeaker up so you can hear it. And so those pulses that you're hearing are the fish using its navigation system, which is based on electrical pulses, to move around in the environment and find objects, and also to communicate with other fish of the same type. So again, let me just remove the fish while you listen. You can hear the clicks very clearly. Take them out. No longer in contact with the water, you can't hear anything because the discharges are not able to go through the air. But in the water, you can hear just fine. Isit here is an undergraduate student, and he and I are going to demonstrate this next week when some freshman students are coming to the lab to learn about what research is going on here at Cornell. And so we hope to demonstrate these electric fish to them, and also to show not just the electric discharges, but to show the electroreceptors which the fish have on the surface of their skin. So we're here to do explorations. Explorations was a program that was started about almost 12, 13 years ago by a graduate student teaching in the introductory biology course, Bio 101. And he thought, what does Cornell do best? Research. And the freshmen never got to see the faculty except for the few teachers that they had in the big courses. So here we are. So now you get a chance to visit a lab, and we hope that this afternoon that you can do a few of the procedures that we do during our research.
The freshmen divided into 3 groups, one to work with Professor Hopkins and the other 2 with Brian and Farah.
And so you're asking what they detect normally. What they detect is other fish. They're very sensitive, and they are tuned, electrically tuned, to the frequency that's characteristic of the other fish. Pretty good? Yeah. Now when you zoom it, you may have to refocus.
Yeah.
The fish is temporarily paralyzed and held in a little plastic holder. The spots on the fish are electroreceptors. Here's a close-up.
Sizes as much, but
You don't see the
I can
difference
tell
in
the
it.
different colors.
You really have to look at it for a while.
Oh, okay.
Because your brain has got to have time to figure out the different categories. And then I want you to try to move it away from, and you can see that the signal is coming from that pore. It really is a spike that's generated from the pore. And notice that the height of the spike is pretty constant. Doesn't vary. It's all or none. There are multiple cells within the organ, and they're probably firing right next to each other, producing 2 spikes, and that's why we see a doublet. Farah Ismail, who's a freshman student, or sophomore student, I should say, that I've known for 1 year at Cornell, and she's been working in the lab for about 2 months. And her project is to work on the same species of fish, but in the larval form. And what we're interested in is how the adult patterns of discharges develop over time in these very early stages of development.
See these things, but generally I'm looking at the interval because I'm looking at SPIs. So once I calculate that, it'll give me like the difference in intervals for all of the data points that. Right. So if you make them more condensed, sometimes it's easier to see what the pattern is.
This is in MATLAB, so if you take CS100M, it'd be very helpful for pretty much any research if you're interested in doing research. So I think the transition from Java into MATLAB is pretty easy because MATLAB's more forgiving than Java. Java's a little bit harder to program. Yeah.
This fish is in Brian's experimental tank. 2 recording electrodes and a hiding tube are on the bottom of the tank. In back, with one end painted red, is a pair of stimulating electrodes. Brian can play back a recording of a male's electrical pulses.
I'm just recording directly the stimulus that I'm playing to the fish. So I want to make sure, that I know what I'm playing to the fish and when I'm playing it. So this just gets directly recorded through the camera. It'll record a certain behavior, and that way you can just do counts of behaviors. So if you see the fish headbutt the electrode or something, you can just push a button and keep track of it.
Here is Brian's recording. Now the playback starts. Watch what the fish does. Now Brian reverses the polarity of the electrode. Where's the fish now? This is good evidence that the fish can detect the polarity of the electric field.
Like our breathing is automatic. You don't have to think about it.
So yeah, I mean the fish discharge. It's under kind of conscious control, if you will. They have descending inputs to modulate the rate of discharge. So let's say, for example, some novel aspect of their environment, like let's say I stick my pen in there and start chasing the fish around, it has a novelty response and it actually increases the sampling rate of that discharge to get a better image of that novel feature. And then similarly, if it's swimming around with boys and girlfish and they want to mate or something like that, they can actively display. Okay. So they're consciously controlling. It's not like an autonomic response. Right.
This is a nice demonstration of bioelectric activity because students don't realize that the electrical activity in their nerves and muscles actually can get strong enough to go outside the animal, and these fishes are just excellent examples of that.